To this point in our tour of the evolutionary forces, we've focused on mutation, which creates new allelic variants, and selection, which changes the allele frequencies systematically based on the difference in fitness that they tend to cause in individuals that carry them. But in developing those models, we have ignored the fact that real populations are structured both in space and they're finite in size. Migration is the process or force that results from or is the movement of alleles between partially isolated subpopulations, and drift is the random sampling of alleles, or perhaps we should say the allele frequency changes that occur from the random sampling of alleles that inevitably occurs during survival and reproduction in any finite population. At the end of the day, migration is a force that tends to make populations less different from each other because they're trading genes and mixing themselves together, and more diverse genetically within each population because they will all tend to have all the alleles that are at reasonable frequencies in any of them. So they're all kept more diverse by trading genes. Drift tends to make populations more different, on the other hand, by allowing their allele frequencies to diverge from each other. And also it makes them less diverse locally, that is, more homozygous, by allowing rare alleles just to be lost, again, by random chance, even if those alleles are not unfavored by selection. The models we'll develop are illustrated by a study of these wonderful water snakes that occur in Lake Erie at its west end on the Ohio and Ontario mainlands, and also on these islands out in the middle of the lake, on which they occur on all of them. And they've been studied by Richard King and his colleagues there, and they show a really interesting pattern of differences in the frequency of bandedness versus unbandedness as one goes from the continental populations out to these most isolated islands. But first, what does mutation do? How does it do it? Here's Heron and Freeman's mouse life cycle diagram again, illustrating migration. So they imagine we have a population initially fixed for the A1 allele. Everyone's a homozygote. There are 800 mice like that. They turn into juveniles and grow up, become adults, but by the time they're ready to reproduce, migration has brought 200 A2 individuals from the mainland onto the island. So now there are 1,000 individuals in all, 20% of which are these A2 homozygotes, 80% of which are now the A1 homozygotes. So the final allele frequencies are P equals 0.8 and Q equals 0.2, quite different from P equals 1 and A2 equals 0, which they were before migration. The model that King and colleagues developed for the water snakes is of the form where one of the two populations, implicitly a continent or core population, is much larger than the focal isolated island population. So migration from the island to the continent has essentially no effect on the allele frequencies on the continent. So we can ignore that. And that could be either because the island doesn't send very many migrants, as illustrated here, or even if it does, just because the migrants coming from the island onto the mainland are a relative drop in the bucket, whereas the same number of migrants coming from the mainland onto the island could affect the island's allele frequencies quite a lot. So the model is really very easy and simple, especially when we can ignore the island's effect on the continent. It looks exactly like our mutation selection balance model, which we recently studied. So we have a term for the allele frequency change caused by migration now, delta Q sub mig, which is just like the term for the effect of mutation. And we have a term for the allele frequency change caused by selection, delta Q sub cell, which is just the same. And for the same reasoning, at an equilibrium, if one exists, these two terms must balance each other exactly, which is to say, delta Q sub mig plus delta Q sub cell, call it delta Q sub I for island, the allele frequency change in the allele frequency on the island per generation must equal 0, which is to say these two have to be equal and opposite in sign. Now back to the islands. The empirical pattern is that bandedness is essentially universal on both the Ohio and Ontario mainlands. Here's a sample from Ontario. All the snakes have pattern D, which is this most strongly banded pattern. The key is over here, D, fully banded. On these islands closer to the Ohio mainland, all 4 patterns occur at intermediate frequencies. That is, there are lots of partially banded snakes, C and B, and also completely unbanded snakes, pattern A, like this guy here on top on the left. And although on the outer islands, Pelly and Middle Island there, the unbanded is most common, the intermediate and fully banded patterns and their underlying genotypes do occur at appreciable, if low, frequencies. So the question is this a result of a migration selection balance in which the unbanded pattern A is strongly favored on the outer islands, but the banded patterns persist due to ongoing immigration from the much larger mainland populations. The argument, the reason to expect that unbandedness is favored out there is that the snakes are hunted by avian predators that see them against the backdrop, which on those outer islands tends to be bare rock, like this rock this collection of snakes is sitting on. The inner islands and the mainland are much more heavily vegetated, and so it's at least a reasonable hypothesis that the banded patterns are favored there because the visual background that the birds are seeing the snakes against has lots of light and shadow contrast in it, and so the snakes blend in better if their outline is broken up by banding. But that same pattern, as you can see, makes them much more conspicuous against bare rock. All right, that's our model. So then here's how it goes. How we put it into equations. Let's let A1 be the dominant allele for the banded pattern, A2 recessive for unbanded. Q will be the frequency of A2 on the islands, the only allele frequency we're thinking about in this simplified model. We're just assuming Q is 0 on the mainland. Then our migration term, delta Q sub mig, is going to be M, the migration rate defined as the proportion of immigrants onto the island from the mainland every generation. Okay, and it's that multiplied by the difference in allele frequency between the mainland, where the allele frequency is 0, and the island, where it's currently Q. Okay, so that's delta Q sub mig, delta Q sub cell. We don't have to write out because we know it. We've seen it before. It's the same as the one we used for the mutation selection balance. The only difference now is that the recessive homozygotes are actually fitter than the dominant homozygotes and heterozygotes. So we'll write the recessive homozygotes fitness as 1 and let the dominant heterozygotes and homozygotes have a lower fitness. So Richard King and his colleagues decided to try to estimate W1, the fitness of banded snakes, and the migration rate by fitting this model against the actual data they got on apparent allele frequencies in the snakes on the outer islands. And they ended up concluding that the fitness of the banded snakes was something around 80 to 90% as high as the fitness of unbanded snakes on the rocky outer islands. And they estimated at the same time that it was most plausible to assume that the migration rate was probably of the order of 1%, somewhere between about 0.3%, 0.003, and say 2.4%. And they got it by the following logic. They just took this model that we just described and plotted delta Q against Q, the current frequency of A2 on the island, for different combinations of M, the assumed migration rate, and W1, the fitness of the banded snakes, which for simplicity they just assumed were all the same. So when you do that, you get curves that look like this. For this case A, the red curve with strong selection, that is a low fitness for banded snakes on the Rocky Islands, and a low migration rate, delta Q is positive under the combined influence of migration and selection until Q becomes very large, almost 1. Only when it gets above about 0.98 or so does delta Q go negative, which is to say, if delta Q is at any of these lower intermediate frequencies, delta Q is positive, so in the next generation, Q will be bigger, and then it'll be bigger again, and bigger again, and bigger again, until finally delta Q gets down to 0, which is, as I said, and as the curve says, is when it's almost already fixed. So this is a bit too high an allele frequency to fit with the substantial frequencies of bandedness on the outer islands, so they don't like this hypothesis so much. Conversely, a weak selection much migration model with, say, W1 as big as 0.9, that is the banded snakes 90% as fit on the rocky outer islands as the unbanded snakes, and a migration rate of over 2% per generation, that produces a curve so low that actually the unbanded alleles are at risk of being lost, and their theoretical equilibrium frequency here, where the curve crosses 0, delta Q of 0, is too low to be plausible. So they like a range of intermediate hypotheses represented by the green curve here, and those are the ones bracketed by these estimates of W1 and M, which are mentioned in the book and in the upper part of the slide. Okay, more symmetrical migration models are also of interest, of course. Everyone doesn't live on an island, almost by definition. So I constructed a corresponding model for two equal-sized subpopulations where selection is working in opposite directions. In island number 1, or subpopulation number 1, the A1 allele is favored. It has a fitness of 1 relative to 1 minus S and 1 minus 2S for the heterozygotes and homozygotes for A2. But the A2 allele is the one favored in subpopulation 2. It has a fitness of 1 there in the homozygotes relative to 1 minus T and 1 minus 2T for the heterozygotes and the A1 homozygotes. All right, so the opposite pattern, similar, qualitatively equal, and opposite patterns of selection, no dominance, just intermediate fitness for the heterozygotes. But I set T to be twice as large so that the strength of selection is twice as high in subpopulation 2 as it is in subpopulation 1. Okay, and then just plotted the equilibrium value of P for different values of the migration rate, a symmetrical migration rate between the two populations, M. So, not surprisingly, when M is 0, when there's no migration, subpopulation 1 fixes for A1, there P hat is 1 for it, and subpopulation 2 fixes for A2, which is to say P hat is 0, but then as we start turning up the gene exchange between the two populations, P hat goes up for population 2 because it's getting an influx of A1 alleles, and subpopulation 1 starts getting lower allele frequencies because it's getting an influx of allele 2 from subpopulation 2. Okay, we just said all that. Above a migration rate of 10%, they both become fixed for allele A2, the one that is favored in population 2 and favored there twice as strongly as A1 is favored in subpopulation 1. So the question for you to consider, it'll be a quiz question this year, it was a clicker question last year, is does a small amount of migration, say M, a little less than 3%, harm both subpopulations, where by harm we mean lower the average fitness of an individual in both subpopulations? And second question, does a large amount of migration with M greater than 0.1 harm both populations in the same sense? Okay, so you'll see that on the quiz. We'll stop now and resume after a break.